coming off that big rock right there.
The rockfish seem to really like to aggregate on the top of these rocks.
We often see that Ophelia sloughing off of the rocks and forming a giant coral beach area just beneath a steep slope.
Yeah, could we drop a target here for Lizzie? She'd like that.
Yep.
When we're done, she'd like to come back and collect some of those skeletons. I'm sorry, Chris, go ahead.
I was speaking a little bit about the management of the area. It's very complex with a lot of jurisdictions. The Navy also has a missile testing range in this area, and the National Park Service manages a large part of the islands and co-manages the waters out to 1 nautical mile. So a lot of groups and entities involved in looking after this area and managing it for all the different uses and purposes.
Yeah, we just have 25 meters left, so I'll add 10, 15 more meters.
All right.
Yeah, we'll stop.
Sounds
good. Is it possible to zoom out of the HiPAC screen just a little bit so I could see C, D, and CE?
Yep, Raj. They're right to the north of us.
And CE is there. I'm wondering, Tom, I don't know if you can look at HiPAC as well, but it looks like a similar feature to what we're on currently, this very complex rocky rubble habitat just to the south of CE. Depending on how we want to continue the survey, it might be worthwhile to head that direction so we can cover that again.
Sure, that's fine.
We were just on?
Yeah, that kind of, right where you're, exactly.
Yeah, it's kind of like north-south. So we can head back to that coral rubble target to sample, and then if you want to go along the axis of this thing, is that what you're thinking?
Yeah, all the way up, like just basically from there up to CE instead of CD. You see that kind of bump just south of CE?
Yep. Yeah, CE is definitely a bigger target than CD, but yeah. If you want to prioritize that, that sounds good. Would be a good angle to get the whole thing.
Yeah. What were those distances?
Just 6 meters, 5 meters.
Roger.
Stopped early.
Okay.
You know, let me know when we're at 200 meters for the transect.
Yep. 196.
Here comes the countdown.
197. If you lose those 3 meters, it can't be compared. 199. Yeah, you're good.
Raj.
Perfect spot to end. Look at that.
And there's some more Placagorgia on that rock right in front of us.
Can we get into position to go back to that coral rubble there?
Yep. So it's going to be 225. Just double check. We'll just come right back.
And would you mind centering up on the DVL screen, please?
Yeah, Raj. Yeah, maybe more like just select the. Yeah, I'm going to do 220.
Roger.
And if for some reason we can't find it, we'll just look a little bit. Lizzie thinks it will run across some other later on.
Yep. Yeah, the coral rubble should make it pretty easy visually.
Yeah, it's bright white.
Yeah. Is that what she wants to sample? Is that rubble itself, or?
Yeah, it's basically the Lophelia that grows on the wall, breaks off, and so it forms this rubble down below. I assume that's what she wants to sample.
I'm just getting blown off a lot by the current, so I'm just coming back slowly.
And when we get back there, Daniel would like us to look for brachiopods.
Oh yeah.
Right around the coral rubble. So.
Roger that.
Sounds like an exciting place to go.
Yeah, 225.
I think more like 220 based on, so not a big difference, but.
Roger. I'm just trying to get enough slack so I can turn around. Okay, that should be good. All right. Yeah, I'm just gonna go right under you. And I'm gonna just follow the DVL screen.
Yeah, the DVL's been super precise.
Yeah, no worries.
Yeah.
All right. Kim, if you want to turn your head as well, I'll meet you at 225.
Yeah, it's only 30 meters back from you. So.
Roger that.
Only. I remember years ago having to do this with Loran and trying to get back to spots. That was not easy.
I won't count my eggs before.
But then in 20, 30 years, just think what it'll be like.
Right? On the shallow water, it's so much easier than the deeper water with the navigation uncertainty as you get deep and deep.
Yeah, and
Roger that. Thank
not
You get
dragging around.
confident.
That and all the tether dragging around and all that.
Right.
Gonna hop
Well,
up a
just
little.
our uncertainty and our pinging. USBL.
True. And we're not in a canyon or anything.
Right. Someone drops a target 2 years ago at 3,000 meters and then expects us to land right on it. So that's not how it works at 3,000 meters. There we go. There's some coral rubble. Lophelia.
Yeah, that's
Definitely
perfect.
looks like it.
The whole field of them, yeah.
Oh, that looks like it. Wow. Goes all the way back. So Lizzie, how much do you want sampled? How many samples? Just grab one of those giant heads of it.
Just start clearing it off.
Might be easier to sit down up here than down below, I think. So,
We have to clean this sanctuary up.
Clean
Hey,
the
this
rubble.
is habitat.
Okay.
Do these branching corals rely on fragmentation for reproduction at all, or seems look like these are all dead.
I don't think so, because they fall down and land here.
Yeah.
And it's not necessarily a desirable place where they end up.
Yeah.
Are the brighter white ones still alive, or are they a different species?
If you zoom in, they're probably all the same species. But if you zoom in,
Nice.
you can see ones that still have polyps. They still will be alive for some of them.
Very morbid over here. Very macabre.
Yes.
All right, looks like we're gonna be all right. Marley, you wanna go ahead and push it on the rubble?
So if you pan to the right to the white colony, there was one that had orange polyps and was alive.
This guy here?
There you go. When we're dead center is.
Nice.
And you can see up the same stalk, there's some with polyps sticking out.
How'd I get down here?
So what are you thinking there, Tom? You want some of the dead and some of the live, or just all dead?
So, Lizzie, do you want the live stuff or the dead stuff? I think she wants to look up for the live stuff above first before we get the dead.
Roger. Marley, you wanna come a little wide? A little more. Okay, roger that.
So we probably have to come off bottom and go up a ways. There's a desmophyllums in there.
Oh, I see. Okay, roger. Okay, full wide there, Marley.
Yeah.
Tom, just so I get a bearing, do you want this colony kind of off to the right here?
Well, first, could we just kind of go up
Sure.
this rock wall
Sure thing.
to take a look?
Sounds like a good plan.
A lot of colorful inverts on that wall.
Definitely are. Because it's coming from someplace, and she'd like to see where it's coming from.
Looks like there's some to the left that's alive.
Yeah.
Yeah.
It does look like there's more alive to the left.
If I get too close, I'm going to get a shadow on the wall for you. Marley, you want to do a partial zoom there? That's great there, Marley.
So those are all cup corals. So if we move up, there's a little canyon to the left of this canyon.
This one here, center screen.
No, keep going to the left.
Roger.
Off screen. So you can start to see some of the live colonies now. And there's got to be more way up on top. But like there's live colonies down in that rubble and on the wall. So if we kind of go vertical for a moment.
Sure.
For a couple meters up.
Road.
Yeah. Could just keep following that canyon, follow all the rubble.
Follow all the rubble, Raj.
So much of it.
Lizzie says it's a Lophelia river.
Very
There's
accurate.
some bigger live stuff.
Wow.
Yeah, so here's the live stuff coming in on the right.
Dead on the upper part.
Yeah.
What's happening?
If we have a chance to
more over
capture any
here.
stills, that would be great.
And these all have like aragonite skeleton up here.
Yes.
Yeah, but it's a much reduced skeleton. If you collect Lophelia from the Gulf of Mexico, for example, less affected by things like ocean acidification, you'll find that the skeleton here is a lot weaker, a lot thinner than what you would see there.
Yeah, poorly cemented. This is amazing.
This is cool.
I
Ah.
know.
It's going right up this pinnacle.
Take us to the peak. Ugh.
No, we
Dinner
have our
relief.
dinner relief. This is gross.
Get out of here.
Get out. Don't want to eat. Go away. Yeah, you want to come back in like half an hour? Okay, fine, Dan. Okay, you can have fun. Yeah. All right. Well, I'm gonna pass over this, guys.
Alrighty.
I'll hold off here.
Hopefully you're having petrolisol for dinner.
All right, don't go too far.
So Lizzie would actually like a photo mosaic of that. She said this would be an amazing outreach tool and a long-term monitoring place for a mosaic.
Are we looking or sampling or what are we doing?
So we're going to do a photo mosaic of the topmost part.
Roger. Okay, give us a second here. We'll pull up our digital still camera. You'll have to re-log in.
What's going on there? Yeah.
No wonder.
It seemed a little dark.
Yeah, that's because I'm not looking at it. I just had the camera zoomed in there.
So Jake, do you know how to take it off of the raw mode and put it on JPEG mode? Do you know how to take it off of the raw mode and put it on the JPEG mode?
You guys want to set it up?
Yeah, because then you could do every 2 seconds, which it says in his sheet there. That's what he recommends.
It's gonna give us a few minutes, guys. We're gonna
Click that.
pimp out the DSC to
Yeah.
get optimal photos here.
You should be able to do 2 seconds when you get to
Sounds good.
Yeah. It does look dark, but is this the feature right here? Is this the height that you want to fly?
You guys are centered on it.
Fly at, Dan? Okay. Yeah, let's see.
You can pull that up over at your station if you want, Steve.
Yeah, I know. I'm already there.
And just take manual pictures.
I would go with that, and I would just set it to go every 2 seconds. Yeah, I mean, it's tough because you're going to be at different heights, right? But that's the autofocus. Oh, I see what you're saying. Yeah, okay. Yeah, I'd give it a whirl.
You gonna be all right with the leash to spin around it, you think?
Yeah, I think so.
Okay.
How far are we away?
12 meters.
That should be funny. Argus is on the uphill side though. Looks like it.
No, don't think so.
What's that?
I don't think so. Argus is back behind Herc. Herc's looking uphill.
Yeah, let me come down and light it up on the sonar.
Yeah. I think that might be it.
Jake, before you
I think
start,
so, yeah.
scroll down so you can, that waveform.
They're 20 meters off the deck here.
Yeah,
I was
down
just
so
freaking out at that.
that's good. Yeah, I just wanted to keep an eye on it as it goes.
What have I got there, 20?
There you go, yeah. You are 15, yeah, oscillating between 15 and 20.
Going. Huh. What did it say? If you go to 10, it lets you do it though?
Just do it manual if you can't set it up.
What's that?
I was saying they could just do a manual like you were saying.
Well, then you might as well.
I suggested that like half an hour ago.
The laser's off.
Okay. Can you put the sub-bottom up there? Yeah, you shouldn't have to do any Argus, so you can be a clicker. Or Steve can bring it up over there and be the clicker. It's up to you guys.
Yeah, what's the well, I gotta do the iris over here and stuff. So.
We get the lasers off.
Okay. Yeah, I'll turn them off. I got it. You ain't got no Argus now. Yeah, ain't got no. Jeez, I actually have to press buttons over here. What the heck? Okay, so we have our video engineer on the video camera, and we have our Argus pilot on the digital stills camera.
We got Mary in the back row on the HD screenshots.
Yeah.
Between all that.
3 people to...
You have to focus at each
I'll
time.
just sit here.
You want to fly the vehicle?
Yeah, we're just sitting here too, so.
Yeah, sorry.
I would just focus it manually right now. Not use the auto sort. Yeah.
Setting up the camera takes a little work because it's different for every. I guess I don't know.
Yeah.
I just use my
And
iPhone
Lizzie
camera.
would like you.
Press the button.
So she'd like you to do, both directions and a few different heights, up to 5 if you can.
Copy that.
That's a lot, but it's a big area.
Yeah, I see now.
You're gonna
She
have
said
to
it may
stay
take an
after
hour.
this.
Yeah, I'm just gonna leave it there, and then now you can snap away
So
at that.
dinner crew is gonna come back and go, what? We got to do what?
That's why they didn't want to leave.
Oh,
They wanted to play
they
with
were
the
ready
nice
to
coral.
sample. Oh,
That's after the 3D mosaic.
yeah,
So they'll
pretty
probably
much.
get to.
Pitches first, then plunder. I give Jess maximum 20 minutes for dinner. That's probably more like 10.
By the time she comes back up, is that what you're saying?
Okay, are you taking pictures? Everything good there? What's that?
I can always move them over.
Yeah, that's all right, as long as they're saving. Or do you want to pimp it now? You're going to have to hand this back over to Kim in a minute, so. Or come back up after dinner and redo the DSC. It'll take them a good. It'll take them a good half hour to go around this thing several times. If they're doing it right.
So if we want, we can go collect first, and then we can let them do the 3D mosaic.
No, if we get the camera set up, it'll stick it on auto now.
Just do the 10 seconds.
They were playing with the, that's all right, just stop it and start it again. It won't actually erase the files. Just says that it does to scare you. They were playing with all the fancy camera settings over there. Just to tweak it for this.
Okay, we're running.
Okay, you happy now? Okay, begin photo mosaic. Yeah. Where do I gotta go to get that half a wrap out? I always go the wrong way. I need to turn the head to the left. Roger.
These fish in the way might be problematic, but.
What's that?
I said all the fish in the way are gonna definitely be hard.
Definitely a little marine snow action there too.
Yeah. Maybe we'll be able to get a tighter one too. That'll clear out some of the water column stuff.
How many different fish are we looking at there, Tom? How many different species of?
Yeah, so we've got widow rockfish, which is a new one for this dive. The kind of orangey red ones are vermilions. The kind of black stripe on the back are either yellowtail or olives. This one near the bottom is a speckled, it's kind of bronzy looking. So that's 4 is all I see currently.
Are we attracting more of them with our lights, or did you guys find them all like this?
We had not gone on the back side, so we were only on this side to the left.
Am I centered up there too? All right.
Yeah. It's great. So many fish.
Most of the fish are vermilions.
You can put it there now if you want, your Argus back.
Yeah, they're not exactly in the brightest light, so might have just been here.
So, did you want just the one in the background, or you want both rocks? That might be.
Just the one in the background with the white coral all over it.
Oh, yeah.
Oh, he's got his mouth at you. He's mad. Oh, there, he's just yawning. because you see this one has all the coral rubble around it. Is it all broken off?
What kind of coral is it?
This is Lophelia. It's a stony coral. It's related to a cup coral. On the east coast, this forms huge reefs. But here, it just grows on rocks and tufts like this.
You're not zoomed in, are you, Steve, or am I that close?
Not zoomed in, as far as I know. I can check. But, oh, I was zoomed in. Must have been left over from the last watch. Did you like where it was, or do you want me to be out like?
Yeah, that's fine. Just probably going too fast for the stills camera.
Got a high shutter speed.
What's that?
The shutter speed's cranked up a little bit, so it should be okay.
So we should be good.
Yeah.
Thing's full of cup corals too.
Yep.
Yeah, the current's got me. Woo!
Current and
Free ride.
pirouette.
Jake, can you scroll down?
You're just used to going fast from
I'm locked out of that
the
guy.
trolley.
Don't worry about it.
Just let it run. It is what it is.
Just tuning in, we're taking a bunch of still images with the idea that we'll stitch them together and make a three-dimensional photo mosaic, just like you see in Google Earth, when you can zoom around and pan around a specific area and check it out in higher detail. That'll allow us to come back to the same spot in the future and see how these corals are doing with different stressors like changes in sea surface temperature and something called ocean acidification. We want to understand the health of the species because it's an important habitat forming species for all the invertebrates and fishes you see living around it. Also, for those tuning in, we are just west of San Miguel Island, the westernmost island in the Channel Islands National Marine Sanctuary. This is a federally protected area, just like a national park in many regards, although we do allow multiple uses. This specific site is in a no-take marine reserve around Richardson Rock in Southern California. The schools of fish you see in the background are a variety of rock fishes, many of them possibly vermilion or sunset rock fishes. I will defer to the guru, Tom, my partner on the science team here, who will tell me if I'm getting any of them wrong. We've seen green striped rock fishes, vermilions, olives, short spined, sword spine, green striped, and also not a rockfish, but we've also seen a ratfish, spotted ratfish, so far on this dive. Once we're done circumnavigating this feature, we will collect specimens of the coral Lophelia that you're looking at now. At the base of this feature, you're seeing dead Lophelia that's grown and fallen off the sides of this rock and piled up down below it. Above, where you see it's a little bit more pinkish, that's actually color within the coral itself, and those are live organisms. They're actually animals. If we, at some point, you'll see we'll zoom in even further as we do our collections, and you'll see a lot more life on that same rock feature, including cup corals. We're going to sample some brachiopods. That's a giant sponge at the bottom left of your screen.
I made it around. I lost the plot there. I've gone around twice now.
I don't know if we got the backside up close because we were far away from that, but
So
there might be a rock in the way.
keep coming to my left till I hit the rock.
Yeah.
And Lizzie asked if you can get as low as possible without crashing on the backside. We have this sponge in the foreground right here.
Heading where? Oh yeah, there's, you can see an Argus. Watch out for that rock. Too late, I crashed. That's as low as we can go there. Just have to skim over the rock here.
That's understandable.
Yeah, that's
It's not
good.
the tiniest vehicle in the world.
That's a good side, shot of the
Oh,
side.
snug up against it with the other side of the vehicle now. Yep, I can see the camera shake. That's some people call it crashing, other people call it landing. You can land with the side. You can land anywhere on the vehicle.
Crash landing.
It's not the cameras.
Could be flying by Braille.
Yeah, totally. Okay.
How many more do you want, Lizzie?
I didn't do an over the top one thing.
Let's do at least that while she's answering.
What's that?
We can get maybe.
I'm waiting for Lizzie to answer, but yeah, we could go over the top. And she said if you could go over the top with the camera pointed a little down.
Yeah, we can't. Go back around for the money shot over the top.
Yeah. Did we get low and kind of
Under that lip there?
I don't know how low we got. I can't remember. Wow, that's so much rubble.
Yeah.
Been here for a while.
Yeah. Contact. I guess I'll go up and over here.
So later on today, in a little bit, when we're done taking the photos of this feature and all the deep sea coral, the Lophelia pertusa that we're recording here, we'll be doing some collecting of the Lophelia and some other organisms. And then we'll be off to another waypoint. We'll collect quantitative data on the abundance of fish and corals and deep sea sponges, so we can better understand the relationship between the fish and those habitat features, and better understand what those population levels are like inside the Channel Islands National Marine Sanctuary.
Not getting any closer, I'll lose the light. Okay, I'm gonna hand it back over to the pro team here, and I'm gonna run away. Thank you very much, Tom. And I'm not sure who else is in there. See ya.
Thank you.
All right. So I hear we did a photo mosaic while we were away.
Welcome back. I hope dinner was great.
Oh yes, very tasty.
So can we still continue some of the photo mosaic? Is that still set up?
Sure thing. Let me know. Sorry, I wasn't.
I think they want to go over the top.
Over the top, right?
We might be, the camera might be facing down a bit. You can double check. I don't know if he tilted it back up. As down as we could go, and then go over the top.
Roger.
And then
Yep, so Lizzie has the direction of going over the top, and if possible, peek into the crevices that are on this side we're on now, and then maybe one more circumnavigation, but with Herc's camera a little up.
Roger.
So more of a horizontal instead of looking down. Thank you.
Sure thing. This is the most down our camera will face right now, and then I'll drive over the top twice, and then we can do the upward facing move that you were saying before.
Roger that.
Great. Sorry? Oh yeah, definitely.
Tom, do we know much about the age and growth of Lophelium?
Sorry?
I don't. I know some people are working on it. But it's hard when they like break off like this to know exactly which colony is how old.
Right.
I don't know why.
So you just want to take a flashlight and shine right in there.
So deep sea corals, we know have the potential to become very old. They can be tens of years, hundreds of years, or even thousands of years old. They can be equivalent to or older than the old growth forests on land. And if they're impacted, if something happens, pollution or perturbation caused by some gear running into them, it's not easy for them to recover from that kind of perturbation. So we want to understand them. We want to know where they are, and we want to protect them as best as possible.
And these deep sea corals grow slowly because they basically are feeding on animals that live in the water column. and they aren't like the shallow water corals that have zooxanthellae in their tissues that they get food from those, which help them grow and to get food. These have to live all on their own in the darkness of the deep sea, sitting in one place, catching particles that go by. So you can imagine there's not a lot of food that way, so they don't grow very fast.
The rockfish will come up interesting in the structure for motion.
Yeah, if they catch a rockfish, they're set for a long time.
Rockfish also are surprisingly older than I think a lot of people realize. Many of them can be decades and decades old. These aren't just 1 or 2 year old fish species that you see out there. And we can tell that because if we look, their bones that grow, their ear bones actually, can be analyzed, and they have rings in these bones that can be assessed for age and growth, just like a tree, the rings in a tree. We can count the rings and find out how old some of these species are.
And that was my first job out of school, was working, doing daily aging of juvenile rockfish. So things that were 6 months old, and you count 180 rings, and you do that over and over and over. You get really good at counting lines in a microscope.
So Tom, would you like me to look up now and try getting on the underside? The shadow might not be very good.
That would be great.
Yeah, we can try.
Yeah, the best you can do. Remember, I only give you the easy things to do.
Roger that.
I also looked at ear bones at one point in my career of little tiny damselfish, and it turned out that some of them were up to 20 and 30 years old as well.
That's interesting. That just doesn't seem right to me, but it is. Just like rockfish being 200 years old doesn't seem right.
And Tom mentioned
Strange
the
things
shell
to
corals
see.
and the difference in growth being much higher. They can grow up to about 1 centimeter in a year. Much more different, much more productive. And you'll see colonies there that are 1, 2, or even 3 meters high. And now you're talking almost 1,000-year-old coral head, even in the shallow waters. So some of them get pretty old too.
Yeah. And they get large.
All important because
Go ahead,
not
Chris.
just because they're animals, but they're habitat forming, and fishes tend to congregate around structure, fish and invertebrates, and so they're home to a wide diversity of marine life. So if you're in the tropics or the deep sea, cruising along a sandy bed, you might see an occasional fish or sea pen or other species. But then all of a sudden, if you see a rock or a boulder or a feature like the one we're looking at now, you'll see a huge congregation of marine life all around it. And corals are part of that habitat forming animals, and it's so important not just to protect in and of their own right, but because they're important for so many other creatures around.
Yep, you can look inside and see little fishies swimming in and out every once in a while, and this Lophelia colonies, or the colonies. Most of them are square spot rockfish, the little ones that have been swimming in and out.
All right, is there any other angles that you'd like, or so we did one up?
I'm asking Lizzie right now. Yeah, so that's good for the mosaic. If you want to try to zoom in some of the holes, but I don't think we have enough light, so
sure. I'll back away a little bit.
Be the only other thing.
Sure thing. Let me get a good spot we can set down a bit. there any spot in particular that she wants? Maybe the one up kind of dead center?
That would be a good one. Try to look in there.
Sure.
She just says she's running back and forth between dinner and this, so she's trying to keep an eye on it and eat.
Actually, I'm going to square my camera and make sure it's all good.
Roger that. And before we leave, if we could put the lasers on it at some point just to see how wide it is. Not to leave them on for long, but just to get a width.
All right, Marley, you want to push in here a little bit, please?
Yep, roger.
That's great there.
Awesome. Thanks.
Sure. Any other holes? One more on the other side?
I don't know if there's another one on the other side. There's a small one, but that's the biggest one right there.
Yeah. If you want, we
I
can
think we
zoom
kind of
in.
looked under this one.
Okay, roger. So you guys all good with the visual inspection then of this?
Yeah, if we could pull back a little bit and put the lasers on just so we can get a size.
Sure.
And then we can go collect.
Sure thing.
That's like 1/2 meter wide there and probably 1 meter wide across this way. Yeah, we don't have to go all the way around. That looks good.
Okay, sure thing. All right, Marley, go ahead and come a little wide.
We're jammed out.
Thanks. All right,
And we
and
can
then
go down to the rubble to collect.
roger that, rebel.
So we're going to look for a live colony. Just anywhere right in this rubble, we can just go zoom it up into it and see if we see a live colony.
Sure thing. So I'll sit down around here and then we can take our pick, I think, of a little bit of live and a little bit of dead.
Yeah, kind of to the right of the lasers up against the rock, it looks like there's a live colony.
Roger that.
So what Alex would like is to collect a big chunk of Lophelia with living tips and dead core, and if there are desmophyllum down there too, grab some of those.
Roger. All right, Ben, go ahead. Okay.
Yeah, so if we go to the right about 30 centimeters, there's some white living rubble right there,
Sure, the one on
right
the
up
rock?
against the rock.
Sure thing.
Not on the rock, below the rock. all the way down in the rubble, right below the lasers now. Yep.
Roger. Sorry, one sec.
Yeah.
I just fell off my parking spot, so I'll one moment. That's better. All right, Ben.
That's good.
All right, Marla, you want to push in there a bit, please? So do we have our pick of any one of these kind of fine-looking colonies here?
I'm going back to Alex.
Sorry, Chris, I don't really hear you very well.
Can you hear me better?
Not very much, no.
Not so much?
Maybe, might be my audio levels here.
So if you go right under the brachiopod, which is to the right, there's that big colony to the lower right.
Sure thing.
That's the one you would like.
Okay.
And then if we could slurp the brachiopod too.
Slurp that brachiopod and grab the coral?
Yep.
Roger that.
And that's just for now. We might come up with more things.
Oh, please do. All right, I'm full to the right, so we can just keep it in view. That's probably gonna be the view we're gonna get. That's all right with you guys? Riley, go ahead and come back, please.
And it looks like a live desmophyllum straight below the brachiopod and the Lophelia, the orangey looking one. So.
So what I'm thinking is that we go for the grab first, stow that, and then if we get kicked off the surface from the suction pump, then at least we have a coral sample already stowed.
Sounds good.
That sounds good.
Great.
That part I leave up to you because I'm a few hundred miles away.
Roger that. All right, Marla, you want to push in there a bit, please? That's great there.
Sweet.
All right. Come away, Marley. All right, someone want to press sample, please? Ben?